So we were just looking at the primary structure of proteins, and that's going to be their amino acid sequence. So if we're looking, what's the primary structure? So you can go through and you can say, oh, it's glycine, alanine, phenylalanine, serine, dot, dot, dot. So looking at how the amino acids are linked together will tell you the primary structure, but that does not actually tell you. So what happens is it's a big string, but remember all of these functional groups that are attached to each of these amino acids can interact in a three-dimensional way, and so that is where secondary structure comes in. So the secondary structure can come in a couple different ways. Now, it's very interesting that it's actually very difficult to predict what the secondary structure will be based on the primary structure. There's a lot of computational models that go into it. It's getting better, but it is very hard to predict because these are all happening in aqueous solution or with water. With different salts, and so all of those will factor into what's the lowest energy state in which these fold into. But we know, for example, that certain amino acids are more likely to form an alpha helix versus a beta pleated sheet. And so using that information, it kind of shortcuts needing to know every little detail. Say, oh, we got a primary structure that's really rich in alanines. It's going to be more likely, based on every structure that we know, to form these type of structures, and that helps make our prediction more accurate. But a secondary structure, so we have two main types. One is an alpha helix. Again, we can get fancier and talk about more, but these are the basic building blocks. So an alpha helix is, I'll show you an example on a modeling program, but it's basically a helix that goes around like a coil, and it goes around in a specific spiral such that the functional groups that we have on the molecule are going to be located right here, right here, right here, and right here. So these are going to have these kind of interaction points, and it could be right here. So we can have like a interaction here, interaction here, interaction here, interaction here. So these are kind of like glue holding our spring together. So we call these they're interactions that happen within the molecule. So we have these within a specific peptide. We have these interactions between the functional groups on each amino acid. So those groups that were in blue right here, or green, these groups are going to interact with each other in very unique ways. And that's going to be why certain amino acids are more likely to form an alpha helix, and also kind of like the coil length. It'll depend on different things. A lot goes into it, but those are how we're going to basically lock and form why an alpha helix forms to have this lowest energy conformation. And so, and then beta pleated sheet is going to be where we have still a continuous. Think of like a continuous strand, but you have your primary structure, but then you have. I'm going to draw this in kind of like 3 dimensions, the best I can. Doing okay with that. Okay, so this is where we have our like NH2 and C double bond. Oh, that's not a C double bond. That is a H C double bond O N C C double bond O N C C. And what we're going to see is that these are going to dangle off here our different functional groups. So these will be our functional groups that we have. And it's going to wrap back around. So we're going to have another. I'm going to draw it this way because I like to draw from right to left, but I'm drawing it backwards from how it's actually going. So we have this kind of wrap around, and this part right here is just kind of floppy. But then we have some structure. So we have our N, C, C, N, C, C, N, C, C. And so we have these carbons have our interactions. And so we have this in blue, we have interactions between the functional groups that are coming off these carbons right here. So those are going to interact with one sheet on top of another. It's called pleated just because these carbon bonds are not flat; they're actually at angles, like we've already shown. And so this type of secondary structure is called a beta pleated sheet. Okay, so we have alpha helix, beta pleated sheet, and that's all secondary structure. But now remember that these are all kind of components of amino acids, so we can actually go a step further into the tertiary structure. So kind of zooming out. And so that's going to be, so we're going to represent the beta pleated sheets. So we could have a beta pleated sheet that looks like this, and then loops around and comes back down, and we have another beta pleated sheet that kind of loops, and then we have a beta pleated sheet like this, comes around, and another beta sheet that comes like this, loops around. And then that structure can then be linked to an alpha helix. That can then have a floppy end and then loop back around. And then you can have some unstructured. And so you'll notice that between alpha helix, we can have interactions. So these can be interactions. And between the beta pleated sheets, we can also see these interactions. It's kind of like glue or Velcro that make them stick together in this shape and just kind of flop around in the solution. So the tertiary structure is specifically talking about the arrangement and the folding of the secondary structure. So how are the alpha helix and the beta pleated sheets arranged together to make a structure? And so this then we can zoom it out even further. Our tertiary structure, we can have this as like one unit, and then we can put those together into quaternary structure. So quaternary structure is as far as you go out. And that is what we call a polymer of proteins. So this would be like one protein. So this is one protein. And we can actually make a bunch of proteins that stick together, and their structures are interacting. So we could have a dimer. So I'm just going to draw shapes that are similar. So this is dimer. That's two. We could have a trimer. Again, I'm just drawing shapes. This is a trimer. Or we could continue to, see how different molecular shapes can fit together in unique ways. So we can keep going on with that, but basically know that a dimer is two proteins. So this is two proteins that once they folded individually, they're going to stick together. and that increases stability. This would be 3 proteins and 4 proteins. And this, again, is all increasing the stability, and so that's why we see a lot of this. And so I will go and show you what this looks like in 3 dimensions. Like, there's modeling programs where we can get these structures, so I'll show you what that actually looks like.